I'm Ki Zhang. I'm Associate Professor in the Department of Biology at Wake Forest University. And my lab used pigeon yeast as the model organism to study gene expression. Gene expression is how the information stored in a gene has been used in making protein. It is a fundamental cellular process and it has many steps. So in eukaryotes, a gene is transcribed into messenger RNA in the nucleus, and then the messenger RNA is exported into the cytoplasm where it can be translated into protein. So in the nucleus, the DNA is packed with histones and the other proteins to form chromatin. So we are trying to understand how the different chromatin structures form and how they affect the gene transcription. And also, once the mRNA is transcribed, they quickly assembled into the core RNA particles and exported into the cytoplasm. And defective of RNA export will cause abnormal accumulation of RNAs in the nucleus. And these abnormal RNA accumulation can form this DNA-RNA structure and cause the mutation in the genomes. We're also trying to understand how the RNA exporting step is being regulated and how the defective nuclear RNA accumulation can cause genome instability. And recently, we also identified a new AAA ATPase in translational elongation and RNA quality control in cytoplasm. And you can see that all these gene expression steps are occurred at the different cellular compartments. However, they are highly coordinated. And our long-term goal is trying to understand each specific steps of this gene expression, how they're regulated, and how they are functionally integrated into the gene expression. All right, let's go to meet my graduate students.
Hey everyone, my name is Fadi Mariotti and I'm a 6th-year PhD student in the Zhang lab. We study things that affect how the genome is compacted inside the nucleus. So you got 6 feet of DNA and you have to really rearrange it and put it inside a very tiny nucleus. And to do that, the cell came up with a lot of different strategies. One of them is to put all the DNA around nucleosomes, and nucleosomes are these beads that are made out of histones. We call them the histone optimers. We see here a quick image of how nucleosomes look like and how the histones are within nucleosomes. And then each histone has a tail, and on the histone tail there's a lot of post-translational modifications that are happening, like methylation, acetylation. I study a specific enzyme that demethylates one lysine on the histone tail of histone 3 at lysine 9 and at lysine 4. My protein of interest is called LSD1. It's lysine-specific demethylated 1. And I study how LSD1 is functioning. We just published a paper about how it really ensures that the chromosomes are really tight and packed around the ends of the chromosomes and around the middle of the chromosomes. These are regions that are called telomeres and centromeres. I'm currently wondering how these proteins are affecting the DNA packing around other genes. So as we know, we have all the genetic information in each one of our cells. So in my finger cell or my eye cell or my hair cell, all the cells have the same exact genetic material, but only specific genes are being transcribed actively in order for them to become the cell that they have to become. And part of this is how genes are being transcribed, and these proteins that I'm studying affect how gene transcription is happening at the right time during the right period of the cell cycle.
My name is Howard Dale, and I'm a 2nd-year PhD student in Dr. Zhang-Reid lab. And what I'm studying now is, the translation and transcription of where two process happens in the cell, but there happens in different compartments. So transcription happens in the nucleus, and translation happens in the cytoplasm. So there's a physical separation of these two processes. So these two processes are really related to each other, and both of them are tightly regulated. So what I'm studying now is we find a very novel protein that which functions may connect these two processes together, and it is a very good tool for us to review what's happening during these two processes and how these two processes coordinate with each other. Yeah, so to our surprise, we also found that this protein have more functions beyond these two processes. Like it could functions in the ribosome associated quality control, which means that the protein needs to be the protein synthesis is wrong, so it has to be corrected, and this protein may function in this way. Yeah, this is very interesting protein, and the more we review it, we feel it is more exciting.
Hi, I'm Jacob Cleary. I'm a second-year PhD student in the Molecular and Cellular Biosciences program, and I recently joined a Molecular Genetics and Genomics PhD program. I'm in the Zhang-Reid lab, and I study how methyltransferases and demethylases cross-regulate and interact with one another to control chromatin states. And broadly speaking, what that means is these proteins that are involved in chromatin, which is the functional unit of DNA, have tails, and these tails can be post-translationally modified, and those modifications dictate whether the chromatin is loose or tightly compacted. And loosely compacted chromatin is available for transcriptional machinery, which means you can produce mRNA transcripts, and tightly compacted chromatin means that it's inaccessible or primarily inaccessible to transcriptional machinery and is likely not transcribed. And I'm interested to see how these important proteins, the methyltransferases and demethylases, interact with one another or interact with each other's complexes to regulate the other's activity.